Part B of Chapter 24, still over the urinary system. The 4 organs of the urinary system are the kidneys, ureter, bladder, and urethra. The star of the urinary system are the kidneys. The structural and functional unit of the kidney is the nephron. There are 2 parts to the nephron, the renal corpuscle and the renal tubules. The renal corpuscle contains the glomerulus and Bowman's capsule, also known as the glomerular capsule. The renal tubules contain the proximal convoluted tubule, the loop of Henle or nephron loop, and the distal convoluted tubule. There are 3 processes that take place throughout the nephron: glomerular filtration, tubular reabsorption, and tubular secretion. Tubular reabsorption occurs mainly in the proximal convoluted tubule, and it's a selective process that includes both a transepithelial step or a step that is paracellular. Almost all organic nutrients can be reabsorbed or reclaimed after glomerular filtration, and we'll see that this involves both active and passive tubular reabsorption, and the 2 routes are transcellular or paracellular. Transcellular route is across or through cells, so we have to cross the apical membrane, the cytosol, the basal membrane, go across the interstitial space, and into the endothelium of the paratubular capillaries to achieve or accomplish tubular reabsorption via the transcellular route. The paracellular route is between tubule cells, so we have to bypass tight junctions, but in the proximal convoluted tubule, they're rather leaky. So things like water, calcium, magnesium, potassium, and sodium can be absorbed through leaky, tight junctions in the proximal convoluted tubule. In this image, we can see the transcellular route and the paracellular route. And again, the transcellular route is through cells. The paracellular route is between cells. Tubular reabsorption of sodium is quite important. It's one of the most abundant cations in filtrate, and its transport is achieved both by primary active transport and by secondary active transport and or facilitated diffusion. Sodium reabsorption by primary active transport is actually able to create energy, which is a means for reabsorbing other substances. So many substances are absorbed with sodium or co-transported with sodium from the filtrate back into the blood or to the paratubular capillaries. Substances that are co-transported with sodium include glucose, amino acids, some ions, and vitamins. The movement of sodium also creates an osmotic gradient for water reabsorption, so water is going to follow sodium via aquaporins. Obligatory water reabsorption is talking about water reabsorption that occurs in the proximal convoluted tubule, which always has aquaporins present. However, aquaporins are not always inserted into the principal cells of collecting ducts, and they're only inserted into the principal cells of collecting ducts if ADH acts on the collecting duct, therefore causing the insert of aquaporins to the principal cells of the collecting duct, and therefore causing an increase in water reabsorption. This type of water reabsorption is not obligatory. It is instead called facultative water reabsorption. The solute concentration in filtrate increases as water is reabsorbed. So basically water leaves and what's left is solute. So that solute concentration increases, and that will cause an increase in the concentration of gradients for solutes. So that's going to encourage certain things like fat-soluble substances and lipid-soluble drugs to either be reclaimed or to stay in the filtrate. There is a transport maximum for substances, and so every reabsorbed substance has a TM, and that just reflects the number of carriers available for the transport or reclamation of these substances. So when all carriers are saturated, any excess is going to stay in the urine. The proximal convoluted tubule, again, is the site of most reabsorption. Almost all nutrients, things like glucose, amino acids, 65% of ions, so things like sodium, also water, are all going to be basically reabsorbed from the proximal convoluted tubule. In the nephron loop, we want to establish a couple of structural differences. In the descending limb, water can leave, so it can move from the tube out into the interstitial fluid of the medulla, and in the ascending limb, water cannot leave, but solutes can. The solutes can leave the ascending limb, but not the descending limb, so this creates a couple of structural differences in the nephron loop in what can leave or enter different parts, so descending or ascending limb, of such a loop. The distal convoluted tubule and collecting duct function in both secretion and reabsorption. However, reabsorption in these areas is mainly hormonally regulated, and antidiuretic hormone, aldosterone, atrial natriuretic peptide, and PTH or parathyroid hormone are going to be the hormones that can regulate reabsorption in the DCT or distal convoluted tubule and collecting duct. Antidiuretic hormone is actually released from the pituitary gland, but acts on the principal cells of collecting ducts. It causes the insertion of aquaporins into these principal cells, specifically on the side of the apical membrane, which is near the filtrate or the lumen of the tube. This will increase water reabsorption into the collecting duct and will decrease the amount of water that's in filtrate. So as ADH levels increase, we see increased water reabsorption and increased filtrate concentration. Aldosterone is also going to target the principal cells of collecting ducts. It'll act also against the distal convoluted tubule. What it does is increase sodium reabsorption, and where sodium goes, water follows. So it causes increased sodium reabsorption and water reabsorption. It's actually really important for little sodium to leave the body because aldosterone, even in its kind of short absence, if it causes a loss of 2% of filtered sodium daily, is incompatible with life. So imbalances in sodium concentration can cause renal abnormalities. So the function of aldosterone is to increase blood pressure by causing salt and water to go into the blood, but because it uses a sodium-potassium ATPase, it also causes the secretion of potassium into the filtrate. And so we get potassium excretion when we get sodium reabsorption with the use of aldosterone. Atrial natriuretic peptide is an antagonist to aldosterone, so it does the opposite of aldosterone. It's going to reduce sodium reabsorption, therefore reduce water reabsorption. It'll decrease blood pressure, and it's going to also affect blood volume. It's released by cardiac atrial cells if blood volume or pressure are elevated. And then last but not least, the parathyroid hormone acts on the distal convoluted tubule to increase calcium reabsorption. Don't forget tubular secretion, which is the opposite of reabsorption, or it's reabsorption in reverse. So we're not going to see stuff be reclaimed by the paratubular capillaries. Instead, stuff will leave the blood to go into filtrate or back to the tube. Almost all reabsorption occurred in the proximal convoluted tubule, and we see pretty much the same for tubular secretion. A lot of it occurs in the proximal convoluted tubule. Selected substances may be potassium or hydrogen ions, organic acids, bases, and even things that are made in the tubule cells, like bicarbonate. We can eliminate undesirable substances or waste products, things like nitrogenous waste, urea, and uric acid, dispose of substances in excess, control blood pH. All these things are possible via tubular secretion. In this image, we can see a summary of the tubular reabsorption and secretion steps. We see that both the distal convoluted tubule and proximal convoluted tubule can perform reabsorption. Most reabsorption in the distal convoluted tubule is hormonally regulated, and even though most secretion occurs in the proximal convoluted tubule, it can also occur via hormonal regulation in the distal convoluted tubule. There's urea recycling between the nephron loop and the collecting duct. We can also see that water can leave the descending limb of the nephron loop and not the ascending limb, and then sodium and other ions can leave the ascending limb but not the descending limb. So before we move on further, we want to establish a couple of different terms like osmolality. And osmolality is the number of solute particles in 1 kilogram of water. So it's the amount of stuff dissolved in a certain amount of water. It does reflect the ability to cause osmosis. The osmolality of body fluids is going to be expressed in milliosmoles. That's a tongue twister, I guess. And then the kidneys maintain this osmolality throughout the nephron loop, starting at 300 and going all the way up to 1200 milliosmoles at the base of juxtamedullary nephron loops. So the kidneys are able to do this via regulation using a countercurrent mechanism. The countercurrent mechanism occurs when fluid flows in opposite directions in two adjacent segments of a tube that has or is connected with a hairpin turn. That's what we see in the loop of Henle, or the nephron loop. We see a descending limb with fluid flowing down towards the hairpin turn. We see an ascending limb with fluid flowing away from the hairpin turn or up. So we have two adjacent tubular segments with opposites or opposition in flow. So we have a countercurrent mechanism. And what we're going to see occur is water and sodium are going to be extracted from different parts of the nephron loop. They'll make the interstitial fluid salty, which causes osmosis out of the loop on the side that's permeable to water, and that water is going to be quickly reabsorbed into the vasa recta, which is of juxtamedullary nephrons. It's called the countercurrent mechanism because there's flow in one part of the loop that's opposite flow in the other part, but we also have the countercurrent exchanger, which is a part of the countercurrent mechanism that also involves that blood flow. So again, the countercurrent mechanism includes the tube and the blood flow. The countercurrent multiplier is another name for the filtrate flow through the nephron loop, or the ascending and descending parts of the nephron loop. It's multiplication because the countercurrent flow enables the effects of the gradient to be increased or multiplied, and the countercurrent exchanger is blood flow in the ascending and descending limbs of the vasa recta. The vasa recta is associated with juxtamedullary nephrons, and juxtamedullary nephrons are going to be the ones carrying out the countercurrent mechanism mostly. And again, we're going to absorb water into the vasa recta of juxtamedullary nephrons, so that water quickly flowing into the vasa recta as a part of the countercurrent exchanger. So it's a countercurrent exchanger that functions mainly in water reabsorption. So to summarize, the loop of Henle, or nephron loop, of specifically juxtamedullary nephrons is going to be utilized in, as well as its blood supply, is going to be utilized to concentrate urine. We're going to be seeing that increasing the concentration of salt in the interstitial space of the medulla will cause water to leave the other side of the nephron loop, but that water is quickly going to be reclaimed by the vasa recta, and therefore will be left with a lot of salt in the interstitial space, and that's going to be used to continue the countercurrent mechanism and pull even more water out of the nephron loop, and this is going to be involved in concentrating urine. So, role of the countercurrent mechanisms is to establish an osmotic gradient from the top of the nephron loop to the bottom. It's going to change from 300 milliosmoles to 1200 milliosmoles. And again, most of the nephron loop of juxtamedullary nephrons is going to be in that medulla. And we're going to see that this allows the kidneys to vary urine concentration. So we're looking again at juxtamedullary nephrons, how they create an osmotic gradient in the renal medulla, and the things they're using to do that are these key players. The long nephron loops of the juxtamedullary nephrons, the vasa recta of juxtamedullary nephrons, that's the countercurrent exchanger, and the collecting ducts that are near or adjacent to the distal convoluted tubule of juxtamedullary nephrons. We can even see here on the right, they're showing us that osmolality gradient of 300 to 1200 milliosmoles. So the countercurrent multiplier is the nephron loop. The countercurrent multiplication in the kidneys is going to be the process of using energy to generate an osmotic gradient that enables the kidneys to reabsorb water from the tubular fluid, also to produce or allow the kidneys to produce concentrated urine. So we're going to use this mechanism to prevent the kidneys from producing liters and liters of dilute urine every day. It's also going to make sure that we as humans don't have to continually drink in order to stay hydrated. We're able to basically reclaim most of that water and use that throughout our circulatory system. So we're going to see that water passes out of the filtrate into the hyperosmotic medullary interstitial fluid, or salt's going to move into the interstitial fluid first, water's going to follow, the water gets reclaimed, the salt stays, so we've made the interstitial fluid hyperosmotic, or we've made it have a higher solute concentration compared to the tubular fluid. In the ascending limb, we don't have water move out because that was only in the descending limb. Instead, the ascending limb was selectively permeable to solutes like sodium and chloride. And we're going to see that in this part, especially that kind of top part of the ascending limb, the filtrate osmolality is going to decrease to about 100 milliosmoles. And between the two parts of the nephron loop, ascending and descending, will be about a 200 milliosmol difference between the two limbs. And that difference is multiplied or will change at about 900 milliosmoles as we go through. The vasa recta is going to be able to preserve that medullary gradient because the salt that leaves the ascending limb is going to stay in the interstitial space, and that water that leaves the descending limb is going to be reabsorbed by the vasa recta. So you can see here the water entering the ascending vasa recta, either from the descending vasa recta or reabsorbed from the nephron loop and collecting duct, is going to make the volume of blood at the end of the vasa recta much greater than it was at the beginning. But that salt's going to stay again in the interstitial fluid, and the collecting duct and nephron loop will use that to concentrate urine. So just a reminder, water can leave the descending limb, but not the ascending limb of the nephron loop. Ions such as sodium chloride can leave the ascending limb, but not the descending limb. The permeability differs, especially in juxtamedullary nephrons. And we're using this again to create an osmotic gradient within the renal medulla so that we can allow the kidneys to produce urine of varying concentration. So the steps here include salt being pumped out of the ascending limb. When salt is pumped out, the interstitial space or fluid surrounding the loop increases its solute concentration or increases osmolality. but the fluid in the nephron loop becomes more dilute as it moves towards the distal convoluted tubule. But now the outside of this nephron loop is really salty, and water wants to follow the salt or move from low concentration to high concentration of solute, so it moves out as well. But it's quickly going to be reclaimed by the vasa recta. But as water leaves the descending limb, it's going to increase the osmolality of the filtrate that remains in the descending limb because this fluid just lost a lot of water, so what remains in the filtrate of the descending limb of the nephron loop gets really concentrated. And that really concentrated filtrate is going to cause sodium to leave in order to dilute that further. We don't want it to stay concentrated in that hairpin turn, so the reason sodium initially left was to decrease the osmolality of the filtrate, but by leaving, it's increasing the osmolality of the interstitial fluid. But when it leaves, that increased osmolality of filtrate entering the ascending limb is going to decrease, and that's what we see is that decreased osmolality as we move up towards that distal convoluted tubule. So again, here's the parts that we're looking at. We have water leaving the descending limb, sodium chloride leaving the ascending limb. We start at about 300 milliosmoles at the end of the proximal convoluted tubule, moving into the descending limb of the nephron loop. It's going to be multiplied at about 200 milliosmoles as we move all the way through the loop till we reach the hairpin turn where we have the most concentrated urine because we just lost all that water. All that's left, you can think of it this way, all that's left is solute, so it's really concentrated or has a high osmolality. Then because of this high osmolality, all this solute is trying to dilute itself. It's trying to kind of reach an equilibrium. As a result, we start to see that sodium chloride begins to leave the ascending limb as the fluid flows around the hairpin turn, trying to kind of regulate this concentration. And on this side, on the ascending limb, sodium can leave, so it does, and it enters the interstitial space. But making the interstitial space salty is just going to make more water leave the descending limb, so it's a back and forth cycle. But as that salt leaves, we're again going to return to a much lower osmolality as we move into the distal convoluted tubule. It's actually even going to be much lower than the fluid or filtrate that entered the descending limb. So we'll reach about 100 milliosmoles. So we see that filtrate entering the nephron loop is going to be isoosmotic to both the blood plasma and cortical interstitial fluid. So that's why it's slightly going to be larger than what leaves the nephron loop. Then water moves out of the descending limb because it can, but salt can't. And that is going to really be water moving down its concentration gradient or osmotic gradient. Then filtrate reaches its highest concentration because it just lost all that water. Then sodium chloride is going to move out because it can in order kind of stabilize the filtrate, and that's going to increase the interstitial fluid osmolality. So it's going to make the outside salty, but the inside more dilute. And then that filtrate that's dilute is going to enter the distal convoluted tubule as it leaves the nephron loop. It's going to be at about 100 milliosmoles. So it's actually hypoosmotic to the interstitial fluid. The interstitial fluid is hyperosmotic or have a lot more solute than the filtrate at this point. And here's our countercurrent exchanger. So vasa recta is preserving that gradient by making sure the water doesn't stay in the interstitial space, taking it right up. So it's going to be reabsorbing water. It even adds some sodium chloride to the interstitial space. So you can see throughout the entire length of the vasa recta, it's highly permeable to water, so water enters. It's also permeable to solutes, so it's allowing salt to go back into that interstitial space if needed. But due to the countercurrent exchanges between each section of the vasa recta and the surrounding interstitial fluid, the blood within the vasa recta remains pretty isoosmotic to the surrounding area. Depending on the osmolality, it's able to take in or release things like water or sodium, whereas only certain parts of the nephron loop could do that. And notice the collecting duct, it would be over here, the loop would be over here. It's allowing water to leave. That water is quickly going to be reclaimed by the vasa recta. So it's going to get more concentrated as far as more solutes are going to be near this bottom portion of the collecting duct. So we're going to see that under the control of the antidiuretic hormone, collecting ducts are going to be able to determine the final concentration and volume of urine. So mainly what we've been seeing here is that juxtamedullary nephrons are used to concentrate urine. We're going to see that the nephron loops and the collecting ducts are going to be able to vary the concentration of urine by using the vasorecta as a countercurrent exchanger or reabsorber of water. And what we were doing is we were basically making the interstitial fluid salty to bring water out of the tubule system, whether it was nephron loop or collecting duct, and making the interstitial fluid salty made water leave, which was great because we want to reclaim that with the vasorecta. So although the loops of Henle or those nephron loops are essential for concentrating urine, they weren't working alone. They worked with those specialized blood capillary networks or the vasorecta, and they surrounded the loops. And so they're equally important or as equally important as the collecting ducts and the nephron loops themselves. The vasorecta capillaries are going to be long, they are hairpin shaped. They're blood vessels that run parallel to the loops of Henle, and their hairpin turn allows kind of a slow rate of blood flow, and that's really going to help us maintain the osmotic gradient required for water reabsorption. So just be familiar with the structures of the juxtamedullary nephron. Don't forget that all nephrons have a nephron loop, but that have a descending and ascending limb, and that the distal convoluted tubule that connects the collecting duct to the nephron loop is going to make the collecting duct very close to or in a similar area as that nephron loop, especially in the juxtamedullary nephrons. They're going to be located in that medulla. Next, we want to look at the formation of dilute or concentrated urine kind of in a different view. What if you're overhydrated or dehydrated? How are we going to change the reabsorption of water? Can we increase it? Can we decrease it? Why would we want to do that? So if you're overhydrated, you're probably going to have a large volume of dilute urine. What your body's going to do is going to decrease ADH production. It doesn't want aquaporins in the collecting duct or distal convoluted tubule. It instead wants to reduce the reclamation of water. So it'll reduce the aquaporins available for water reabsorption. So ADH will reduce that. ADH will reduce that if it's reduced in its production. And so the urine will really be about 100 milliosmoles. It'll be very dilute urine that is produced in someone who is overhydrated. If someone is dehydrated, we're going to try to reclaim all the water we can. ADH will be maximally released. We'll put as many aquaporins as we can into the distal convoluted tubule and collecting duct. In severe dehydration, we'll actually reclaim about 99% of the water that's actually trying to get through the filtrate. Instead, it'll be secreted into the interstitial space, and we'll clearly reclaim that using our countercurrent exchanger or vasorecta. So you can see here that if someone's dehydrated, they could have urine that's about 1200 milliosmoles or has a lot of solute compared to water in it. So here's that process. We can see the mechanisms for forming dilute or concentrated urine. So for forming dilute urine, that mechanism's on the left. For forming concentrated urine, that mechanism is on the right. So if we have a decrease in osmolality of extracellular fluids, we decrease ADH, we decrease the number of aquaporins, and we decrease H2O or water reabsorption. That's going to lead to a large volume of dilute urine. So if you're overhydrated, we're not going to reabsorb water because we want that excess water to leave the body because we have enough. You're overhydrated. You have plenty of water. Whereas if you're dehydrated or we have an increased osmolality in the extracellular fluids, then we're going to increase ADH, which is going to cause more aquaporins to be put in the collecting duct, which is going to increase water reabsorption, which means we reabsorb most water, so it's not going to remain in the urine, and you produce a small volume of concentrated urine. So if you're dehydrated, we're going to take as much water as we can because you don't have a lot to begin with, and so we can reclaim almost 99%. But again, it's going to be antidiuretic hormone regulating that at the distal convoluted tubule and collecting duct. Next, we're going to look at urea recycling and the medullary osmotic gradient. So with urea recycling, we're going to see that this molecule, which is a nitrogenous waste, is going to be used to help us regulate that osmotic gradient that we were creating using the countercurrent mechanism. So urea recycling in the inner medulla, it's also going to contribute to the osmotic gradient that we were generating in the nephron loop or loop of Henle. So we'll see that antidiuretic hormone increases water permeability because we can add aquaporins to the collecting duct, for example, but that doesn't increase the urea permeability, especially not in the cortical or outer medullary collecting ducts. So that can cause urea to concentrate in the tubular fluid in that segment of the nephron. In the intermedullary collecting ducts, it increases both water and urea permeability, and that's going to allow urea to flow passively down its concentration gradient and into the interstitial fluid. So we're going to see the secretion or passive flow of urea down its concentration gradient into that interstitial space, and that's going to add to the osmotic gradient that helps drive water reabsorption. So as filtrate enters the ascending limb of the nephron loop by facilitated diffusion, the cortical lectin conduct is reabsorbing water, again, but not urea. So that urea in the deep medullary region is not going to be highly concentrated. So it's making the interstitial fluid, we'll say, even more salty, as an example. But it's going to make that even more prone to cause water to leave the descending limb. So it'll increase water reabsorption by making the interstitial fluid of the medulla have a high osmolality. All right, and then next we're going to look at diuretics. And so in the next part of the urinary chapter segments, we will start with diuretics and finish out part B of the urinary system.